1. Recombinant DNA is directly injected into the nucleus of an animal by a method called dash. A. Microinjection. B. Biolistics. C. Heat shock. D. Disarmed pathogen vector. Answer A. Microinjection. 2. Crystals of Bt toxin produced by some bacteria did not kill the bacteria themselves because A. Bacteria are resistant to the toxin. B. Toxin is immature. C. Toxin is inactive. D. Bacteria encloses toxin in a specific sac. Answer C. Toxin is inactive. 3. Name the interaction in which one species is harmed and the other is unaffected. Answer Amensalism. 4. Fruit developed without fertilization are called dash. Answer Parthenocarpy. 5. Select the correct pair. Answer 1. Plant cell cellulase. 6. A. Expand ELISA. Answer enzyme linked immunosorbent assay. B. Write down the basic principle involved in this technique. Answer antigen antibody interaction. 7. Two characteristics of population density is given below. Explain the terms. A. Natality. Answer natality refers to the number of births during a given period in the population that are added to the initial density. B. Emigration. Answer emigration is the number of individuals of a population who left the habitat and gone elsewhere during the time period under consideration. 8. Observe the given figure of microsporangium and label the parts A, B, C and D. Answer A. Epidermis. B. Endothelium. C. Middle layer. D. Tapetum. 9. Agrobacterium vectors are used to introduce nematode-specific genes into the host plants. A. Name the nematode which infect roots of tobacco plant. Answer Meloidogyne incognita. B. Define RNA interference. Answer silencing of a specific mRNA due to the complementary dsRNA molecule that binds to an and prevent translation of the mRNA silencing. 10. Observe the figure given below. A. What are age pyramids? Answer if age distribution % individual of a given age or age group is plotted for the population, the resulting structure is called an age pyramid. B. Identify the type of age pyramid labeled as A and B. Answer A. Expanding. B. Declining. 11. Amplification of gene of interest is done using polymerase chain reaction. Name the enzyme used in PCR and write the characteristics feature of this enzyme. Answer the enzyme used in PCR is Taq DNA polymerase. The characteristic features of this enzyme are it remains active during the high temperature induced denaturation of double-stranded DNA. 12. Fill in the blanks with type of interaction observed among the following species. Answer fig tree, wasp. Type of interaction mutualism. Orchid, mango tree. Type of interaction commensalism. Cuckoo, crow. Type of interaction parasitism. Abundant tortoise, goat. Type of interaction competition. 13. Water pollinated flowers have many peculiarities. Write down any 4 such peculiarities. Answer pollen grains in many such species are long, ribbon-like. Pollen grains are protected from wetting by a mucilaginous covering. Flowers do not produce nectar. Flowers are not very colorful. In seagrass, the flowers remain submerged. Write any four peculiarities. 14. Explain the method of genetically engineered human insulin production by Eli Lilly in 1983. Answer. Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them into plasmid of E. coli to produce insulin chain. Chain A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. 15. Explant and totipotency are two terms related to tissue culture. Define these two terms. Answer. Tissue or plant part used for tissue culture is called explant. Or any part of a plant taken out and grown in a test tube under sterile conditions in special nutrient media in tissue culture. The capacity to generate a whole plant from any cell or explant is called totipotency. 16. Observe the figure given below. A. Identify the population growth curve A and B. Answer. A. Exponential growth or J-shaped curve. Logistic growth or Verhulst-per logistic growth or sigmoid growth or S-shaped curve. B. What do R and K stands for in the equation given in the figure? Answer. K. Carrying capacity. R. Intrinsic rate of natural increase. 17. EcoR1 is an example of restriction endonuclease enzyme. A. Name the bacteria from which EcoR1 is isolated. Answer. Escherichia coli. B. Explain the naming procedure of EcoR1. Answer. First letter, that is E, represent the first letter in the genus of the bacteria from which the enzyme is derived. The second and third letter, that is Co, represent the first two letters from the species of the organism. The fourth letter, that is R, represent the first letter of the strain of bacteria. The Roman number 1 represent the order of isolation. Or E represent Escherichia, Co represent coli, R represent RY13 strain, 1 represent the first order of isolation. 18. Predators act as conduits for energy transfer across trophic levels. Write any 3 roles of predators. Answer. Predators reduce the intensity of competition among competing prey species. Predators help in maintaining species diversity in an ecosystem. They keep prey population under control. Used in biological control or pest control methods. Write any 3 roles. 19. Transgenic rats, pigs, sheep, rabbits, etc. have been produced by rDNA technology. A. What are transgenic animals? Answer. Animals whose DNA is manipulated to possess and express a foreign gene are called transgenic animals. B. Write 2 benefits of transgenic animals. Answer. Used to study the normal physiology and development of a gene. Transgenic animals, that is mice, are used in testing the safety of vaccines. Used to understand the role of a gene in the development of a disease. Transgenic animals are used for the production of biological products. They are used for toxicity or safety testing of chemicals. Write in it 2. 20. Observe the figure given below. A. Identify the instrument in the figure. Answer. Simple stir tank bioreactor. B. Label the parts A and B. Answer. A. Motor. B. Sterile air. C. Write any one use of this instrument. Answer. Bioreactors provide optimal condition for large-scale production of products. Bioreactors are commonly used for continuous culture or cell culture. Write in one use.